Okay, so next speaker is Professor Arnab Bhattacharjee from Jawaharlal Nehru University in New Delhi. So he's going to tell us about search and probably search and capture. Let's wait and see. Okay, Arnab.
Thank you, and thank the organizer for inviting me in this workshop. And I'm first time here at ICTS. It's a nice campus. Fantastic workshop is going on, and I met several colleagues who had GNU connections. Maybe at the capacity of student, at capacity of faculty, right? And so this picture is from the highest point of GNU. We called it PSR, and this is to refresh their memory. And for others, this is to invite them in GNU for maybe a talk and explore the nice campus. Okay. So today I'll speak about a search problem. So search in general is a common phenomena in biological system. And if you, for example, consider animals search for their prey, yesterday Shakuntala mentioned about this running and tumbling motion of E. coli searching for their foods. And here I'll speak about search of proteins on DNA. And so definitely my system of interest is the DNA and the whole genome. And this is a nucleus of a cell. And it's a fascinating system of interest because it contains the whole set of DNA, for example, for human, and it controls nearly everything of a walking and talking person. And how does that happen? As Stephen already mentioned, that during in central dogma, that whatever be the DNA metabolic processes, be it transcription, be it replication, be it DNA repair, everything is basically triggered by binding of a protein on a DNA, and this binding should be very specific at a very specific position. Now, if you consider human genome, it has almost billions of base pairs. So then question is how a protein finds its specific site out of that huge combination. That remains a challenge to understand. Particularly if you consider this multilayer structure of chromosomal DNA, this is even more difficult because what we start understanding at the very basic level, DNA is a double helical structure which then wraps around 1.67 times on histone octameric protein, which forms these nucleosomes. Then they combine together to form this 30 nanometer fiber, which goes several other compaction to fit, this is almost, if you stretch it, is 2 meter long thread, which fits into nucleus, which has a diameter of micrometer scale. So it's a huge compaction. And one of the major key component of this compaction is this called TAD, or topologically associated domain. So this is basically a segment of DNA sequence that physically interact within it, and therefore inter-TAD interaction is very rare. And therefore, if you consider about expression of genes, which is triggered by expression of other genes, then it happens only within a TAD and not inter-TAD interactions. So given this multi-layer structure, at each level, the structure regulates the accessibility of DNA sites, right? So with this structure, one can really ask this question that how do proteins search their specific sites on chromosomal DNA? A bit of history. The problem was realized fast almost around 50 years back, when Riggs published back-to-back 3 papers in Journal of Molecular Biology characterizing the binding of lac repressor protein on DNA. So lac repressor basically is a protein that controls the metabolism of lactose, and if this protein binds at this operator site, it prevents the movement of this RNA polymerase and thereby hinder the expression of the gene. So what he found is interesting because he said that on this 50 kb DNA, the association rate of this repressor protein is 7 into 10 to the power 9 mole inverse second inverse. And he argued that this is almost 10 to 100 fold faster than the upper limit estimated for diffusion control process, three-dimensional diffusion control process, which we call Smolchowski limit. And it was not understood that how really protein achieves this speed up. So almost around 10-11 years after, Berg and Hippel, they proposed a mechanism. They said, okay, see this protein, they do not really do only exclusively three-dimensional diffusion and encounter its specific site. Rather, it reduces its search space. So what it does, let's say this is the operator site where the protein needs to bind, the green is protein. So instead of finding this through three-dimensional diffusion, it may associate at a site on the DNA far from it and then can move along the DNA, and essentially that will be a one-dimensional movement because it is on the DNA. And there are different types of movement. If it is moved like this, they can call it sliding. It can perform small jumps on the DNA, which is hopping, and even if two sequentially far segment of DNA are nearby, then the protein can jump from one segment to another segment, which is called intersegmental transfer. So this is the idea that they said, okay, they termed this as facilitated diffusion, and they said this is the way that protein really speeds up their search process. In 2012, Berg and Elf established that this phenomena can be observed in living cell, and typically this lac repressor protein, and it takes almost 3 to 4 minutes to find their or recognize their specific site. So a very interesting observation they reported is that even though the protein slides over many times on its specific site, it takes a while before it binds, right? So that means even if it is passing through this site, it cannot recognize exactly. So then question arises, what actually controls this search process? So if you consider it, you will find quite a bit of similarity between real life traffic and transport phenomena. So for example, if you book an Uber and you want to move from one place to another place, you will find that this kind of picture you will see where some portion is red, which signifies obviously the crowding or congestion, right? So similarly, within nucleus, you have multiple copies of many other proteins which act like crowders, and they kind of may pose a challenge to this protein which is moving on the DNA. So how do these proteins search? We looked into this problem quite a bit, and what we find, if you have an experience to travel in crowded bus or crowded train, probably you know that when you want to get down at a certain stop, there is a specific way to do that, and the way is you need to pose yourself close to the gate within the crowd. You need to identify the crowd that will get down at that stop, and then you need not to do anything. You basically, when the stop comes, the crowd will take you out of that bus or train and you will be at your destination. So similar thing we observed here is that when these crowders are there, these form a depleted volume around this DNA. And when the protein, searching protein, is within this depleted volume, it experiences a microviscosity that is much lower compared to the bulk viscosity. And then the protein can move much faster, and even if the protein has some sort of weak affinity with the Crowder molecules can carry this protein along with it, making the search faster. The second way is that if you can see here, there is another road possibility, but it will take probably longer time, and then that is why Google didn't suggest you. So it could be because of road closure. Similarly, within cell you can have road closure. That means many sites can be occupied by already other proteins, right? So when a protein needs to move onto it, it has to find out a way to bypass such obstacles. The third is that like speed breakers and potholes on road, which reduces the search speed. On DNA, similar kind of situation may arise. For example, let's say this is the target site where the protein needs to bind, and basically this is a combination of ATGC. So there are many other sites, this kind of, not exactly but close similar sequences can arise. So we call these sequences as pseudo target sites. And now when the protein goes through these pseudo sites, it can get trapped there. right? Now, if the protein is very fast, then it can pass through these pseudosites very easily, but then it can also miss the target site because this is very fast. On the other hand, if the protein has high affinity for this for its target site, then the protein can also get trapped for longer time on this pseudosites. So there is a speed stability, it's called speed stability paradox, which says that if the speed is very high, then it can miss the target site many times before it actually binds, and that can lengthen the search time. On the other hand, if the stability is very high, then also you can get trapped into local minima, into pseudosites, and then also your search time will be longer. So question is how protein balance between these two feature like speed and stability. So protein has actually different way of transport. It has fast and slow mode of transport, right? What is that? Okay, one is called sliding, as I already mentioned, where the protein moves along the DNA. In 2009, there was a fantastic work by Biman Bakshi and Sanizi's group. What they did is that they measured the diffusion coefficient, one-dimensional diffusion coefficient along the DNA of many proteins, and with respect to 1 by r, considering that this is diffusion given by Einstein's equations, and they found the correlation is only around 0.52, right? And so what then they proposed that the protein doesn't actually go just one-dimensionally. Instead, when it goes, it also rotates around the DNA. And when it rotates, this kind of equation appears, which shows a fantastic correlation. Computationally, through simulation, it was first shown by Levy's group from Weizmann, and he did this kind of simulation where this protein moves along around this DNA major groove nicely through a spiral kind of motion, and while doing so, it can also go up and down along the DNA. Yes. These are experimental, yes, single molecule, yes. No, in vitro, yeah, right, okay. So this is, and as you can see, during this motion, the protein reads each and every basis, so essentially this is a slow motion, slow dynamics. In comparison, a fast dynamics where the protein if the protein has two DNA binding domains or a tail that has a binding patch, for DNA binding patch, so and if it senses to another segment of DNA nearby, it can transfer from one segment to another segment, which we call as intersegmental transfer. And this kind of motion is very effective for bypassing the scanning of a huge section of DNA. So this essentially is a very fast kind of motion, which was also supported by Clore's group later on. And it was also, if you find the free energy profile for this, you will see that this kind of transition state you can observe where these proteins can form this kind of bridge complex between these two DNA before it goes into one of the DNA segment. Now, the question is, this fast, slow, different transport modes are there. Who controls that? What needs to be chosen, right? How protein selects that? Well, there are many factors that governs that, but the major factor is obviously the electrostatic interaction and therefore the salt condition. So, for example, if you have a very low salt condition, there is no screening effectively, and then the protein can see the DNA very effectively. So it's a patchy surface, the protein will stick on the DNA and do mainly the sliding dynamics. So its sliding propensity will be highest. The opposite, if the salt is very high, then it will be a huge screening effect and the protein will not be able to see the DNA, and then we will have a huge three-dimensional diffusion. At intermediate salt, the protein can sense the DNA. It can, but it is not that sticky, so it can perform small jumps, which we call hopping, and therefore that is maximum at intermediate salt condition. Similarly, intersegmental transfer is also dependent on salt condition. But whatever we have shown so far is basically on short segment of DNA. But as I already mentioned that chromosomal DNA is much more complex, which has a higher order organization as well as supercoiled topology, many bends, nicks. So really this doesn't explain how proteins make it on this kind of complex structure. So I'll try to explain that. I'll try to answer that by using simple model and computer simulations. Yes. It depends on how high salt means how high you are considering, right? If it is very high, definitely, obviously there is a conformational change, but typically up to around 200 millimolar, there is not that much of change. No. So this is the nucleosome structure, where around 150 base pair of DNA wraps around histone octameric protein. And as because it wraps around histone octameric protein, so all the sites of the DNA are already occupied by the histone proteins. So then question is how other proteins which are non-histone protein can access these sites. So fortunately this structure is not a static structure, rather it's a dynamic one, and it was shown that from one end of the DNA it can transiently unwraps and rewraps. It is known as this kind of dynamics is called nucleosome breathing dynamics, and in 2018 the corresponding structure has been found out using cryo-EM technique. So if you see this kind of structure where this is the transiently unwrapped segment, and one can consider this as kind of linear DNA, right? So if you consider this kind of portion as linear DNA, where this is, let's say, the target is located at mth site, this is the beginning and the end, and then the protein can move on this segment randomly, right, which is a one-dimensional random walk, and you can ask when the protein will for the first time reach at this site, so you can recast your problem into first passage time problem. And Kolomeski from Rice University has done a lot on this, and you can simply write the time evolution of the probability is that from the nth state it can on the using one-dimensional movement it can move to n plus 1th site or n minus 1th site on the DNA, or it can dissociate off from the DNA with a rate k off. Similarly, this is basically the boundary condition at 1th site. From 1th site it cannot move this side, so it has to move on to a second site. Similarly, from lth site it cannot go from this direction, it has to move on this direction, and this is the solution phase f naught, which tells that with equal probability that protein can come on any site on the DNA. And then you can use a Laplace transformation to make these equations simpler and solve them. You will find FNS, and from that you can find out the mean first passage time, which I'll not speak about the details because that is already discussed in quite details. So if we consider about the nucleosome structure, then you can see we have considered here one fully wrapped structure where the full protein is wrapped on the nucleosome, and one state which is partially unwrapped structure where the part of the DNA is unwrapped from the nucleosome. And then these two structure is connected, conformation is connected by these two rates, closing and opening rate. And the protein can come on it with a rate k on, and it can dissociate off from it with a rate k off. Now, if you see these transcription factors, there are mainly two different kinds of transcription factor. One is called pioneer factor, which has a higher affinity for the nucleosome, and it can bind even when the nucleosome is in fully wrapped state. And then the protein can bind this red one, and it waits until it opens, and as soon as it opens, it starts this moving one-dimensionally. On the other hand, some other proteins which are called non-pioneer proteins, which cannot really bind with when the nucleosome is in fully wrapped state. It waits until in the solution phase, wait until it opens, and then it binds to do this kind of movement. So again, you can write a simple equation for this. So this is basically for the fully wrapped state, the segment that can unwrap. So what can happen? Obviously, if the protein is on it, it can dissociate onto the solution phase. This is one possibility. Another possibility is that this segment can get unwrapped, right? For the rest of the part, obviously only one option is there, that it can be, the protein can get dissociate from it, because the other segment we didn't allow it to unwrap. So this is kind of asymmetric nucleosome breathing. For the partially unwrapped state, in addition to this, you have also one-dimensional movement on the DNA, which is with rate u, and then you have these boundary conditions. If you solve this, you will find the first passage time, and what you need as input, you need several kinetic parameters, kinetic rate constants. So here we have rate constants for 3 proteins, REB1, CBF1 and LEX-A. So these first 2 are pioneer factor proteins. So, and this one is non-pioneer, and we have rate, off rate and on rate, both on nucleosome and on linear DNA. So these are the experimental results. So with this, we calculated the target search time for both, pioneer factors and non-pioneer factors, both on linear and non-linear and nucleosomal DNA. And what we found is, if you see on linear DNA, the non-pioneer factor is much faster compared to the pioneer factor. But on nucleosomal DNA, the pioneer factors are, or the search time is order of magnitude lesser compared to the non-pioneer factor. Another most important thing here is that the search time here, if you see, that is inversely correlated with the affinity of the searching protein. That means the protein that has highest affinity searches it the fastest. So this is kind of, against the speed stability paradox, which says you need to maintain, because if you have very high affinity, you cannot move faster. But here we are showing that on nucleosomal DNA, the principle is slightly different, and you need really a high affinity for the DNA. So why this is so? Well, we found that what happens when the nucleosome opening rate, you change it. So this is the target search time, and we see that for pioneer factor, the search time goes down if the nucleosome rate increases. So what happens? So with the nucleosome rate increases, that means the pioneer factor, because of it has high affinity, it remains associated with the fully wrapped conformation. As soon as it unwraps partially, it starts one-dimensional movement to search for the target site. And therefore, if the unwrap rate is faster, then basically the protein has to, the waiting time for the protein is lesser, so it searches faster. Now, if you do the same thing for non-pioneer factor, we found that this is a non-monotonic behavior, and the fastest search happens at a intermediate opening rate. So why this is so? This is because this opening rate, because of equilibrium constants, this opening rate, if it increases, the closing rate also increases, and if it decreases, closing rate also decreases. Now when opening rate is very low, that means the closing rate also very low. Now most of the segment, the unwrapped segment, remains open for longer, and this non-pioneer factor is trying to search on that protein. But if it goes into wrong direction, then there is no one to say, hey, you are going wrong, you need to correct yourself, right? So that means, but as soon as it rewraps back, with rewrapping, as I said, non-pioneer cannot remain associated with the nucleosome, it has to get off it and goes into solution phase. So that is basically kind of resetting, right? As whenever it falls, the structure rewraps, it goes into solution. So that is resetting. Now if the resetting is very slow, then the protein can go wrong for longer time and your search time is higher. But on the other hand, if the resetting is very high here at this region, then what happens? Every time protein tries to move, resetting happens, it goes into solution phase. So that again increases the search time. At intermediate condition, you get the optimal search time. For pioneer factor, obviously with this rewrapping, the protein doesn't get dissociated. Instead, it has its own rate of dissociation, and you see similar kind of structure. What is important here is that if you see this on rate, if you increase the on rate with this color, the minima shifts towards right? So higher the on rate, higher the off rate as well. So we found that if you plot a contour like with resetting rate and the on rate, you will get this kind of structure where this red line corresponds to the fastest target search time, so which says there is a correspondence between this on rate and off rate. And this black line is basically 10% deviation from that fastest search time. So if we see this, both the pioneer factor falls within this region and the non-pioneer factor is located here. So what we conclude is that the quickest search is observed when the association rate is approximately compensated by the dissociation rate or the resetting rate. For non-pioneer factors, the association and resetting rates, they differ by almost 5 orders of magnitude. And therefore, it times increases by 80 times compared to pioneer factors. Whereas for pioneer factors, their ability to invade nucleosome is originated because of this association compensated dissociation mechanism, which allows them to, there is a trade-off between mobility on the nucleosome as well as making wrong step or error on the nucleosome. So that is, so this mechanism basically helps into finding the search target faster. But this is for one pioneer factor, one transcription factor, but in reality you will have multiple transcription factor, and that is much more complex situation. So here we have considered and here we have taken computational approach. So here we have two factors. These are pioneer factors, SOX2 and OCT4. They are pluripotent transcription factor, which means they convert the somatic cell into pluripotent stem cell. Now they bind in this entry-exit region of a nucleosome. Now if you want to study their search process, what we did is, okay, we have two structure. One is fully wrapped, one is partially unwrapped, and then you can construct a free energy basin corresponding to this structure, which is basin 1, another basin this corresponding to this structure, and these two are separated by energy delta. So obviously higher delta means the basin 1 will be more stable compared to basin 2, and then you can have a coupling constant which can connect these two basin, and this is related to the free energy barrier. Now you can construct from this two basin model, you can get a smooth potential function by the eigenvalue of this characteristic equation will give you this energy for this two basin model. And you can obviously have one reaction parameter, which is we call chi, which is basically the ratio of these two eigenvectors. With this, you can have a nice two-state model where you can see with chi, this is time step evolution, you can see this nice two-step model, this is the bound fully wrapped state, this is partially unwrapped state, and by changing this delta and gamma, you can change the free energy barrier between these two states, and you can match it with the experimental results. And also we have seen that when it does this, there is a bulge kind of formation within this DNA, the unwrapped loop, unwrapped segment. So this is a small movie showing this wrapping and unwrapping dynamics of the protein, of the DNA on histone protein. Okay, now coming to the search process. So we have looked into the diffusion of SOX2 and OCT4 separately on this nucleosomal DNA, and we found that OCT4 is the diffusion on the nucleosome is almost two-fold faster compared to SOX2. And why this is so? We found that the movement of SOX2 is dynamically coupled with the bulge that I have shown with that, and it is almost 85%. So what happens, SOX2, the size fits very well with the bulge size and it goes into that, and with the movement of the bulge, the SOX2 also moves. So basically it is restricted movement, and that's why it is slightly slower compared to OCT4. Now if you come to the kinetics, what happens, SOX2 is faster compared to OCT4 because although the diffusion of OCT4 was faster, right? So why is so? Okay, we again from our simulation calculated the on and off rates from the simulation trajectory for SOX2 and OCT4, and you can see for SOX2 this on and off rate are quite comparable, whereas for OCT4 it doesn't. So again the principle that we established from our analytical model, that association compensated dissociation, that again holds on this simulation as well. So this is a small movie, doesn't matter, it shows how they move towards this target site. and eventually they bind. Okay. Now, what happens when both the proteins are present in the system? You can see that SOX2 is still faster, and this result is in accordance with a live cell experiment, which also says that SOX2 moves much faster compared to OCT4. But it also says that the binding of OCT4 is cooperative. So that means binding of SOX2 facilitates the binding of OCT4. So to understand that, we calculated the thermodynamic parameters, and for the free nucleosome, the nucleosome bound with SOX2 and nucleosome bound with SOX2 and OCT4. So what we found is that if you see the net change in enthalpy for binding of OCT4, it is positive. So that means it is anti-cooperative in terms of enthalpic contribution. But if you see entropic contribution, this actually overpower, it's a positive entropic contribution, it overpowers the enthalpic anti-cooperativity, and therefore this cooperativity actually originated because of entropy, change in entropy. So how does that actually happen? So to understand, we have a model of this is where we have done information transfer landscape of the unwrapped segment. So what we have done, we have calculated the fluctuation of measure groove width of the DNA, and using the Shannon entropy formulation, but with time delayed probabilities of the measure groove width fluctuation, we found out the net entropy transfer. When there is no protein, only the nucleosomal DNA, there is net transfer is 0. But when SOX2 binds, it gives entropic funnel here, creates an entropic funnel which is entropic sink, and this portion which is deep inside the nucleosome, here entropy is higher. So if the protein, the OCT4 is close to this, it can sense this entropy difference, and then it moves along this direction, and after OCT4 binds, you can see that this sink disappear. So we conclude that this faster binding of SOX2 obeys association compensated dissociation mechanism, and specific binding of SOX2 facilitates the target search of subsequent transcription factor in a cooperative fashion, and specific binding of SOX2 also generates an entropic sink that creates an entropic funnel around it. The following transcription factor senses the entropic funnel and moves towards it, and through this entropy transfer mechanism. So this cooperative phenomenon can also be observed on DNA through DNA shape complementarity. So this paper tells that these are the two proteins. It's a FIS protein, it's a GIS protein, and they suggest that FIS protein, when binds on DNA, it deforms the DNA quite a bit, and because of that deformation, this GIS is a minor groove binder. Because of this minor groove is wide open, and it is suitable for binding of GIS. So GIS comes and bind. Okay. So to understand this, we again did the kinetic study, and what we found is that when GIS is present, this is the kinetics, this is the number of native contacts that form with time, and when FIS is present, this line, black line, this is much faster, the kinetics. And we also check that, okay, let's say there is no FIS, but we artificially constrain this angle, and therefore we get almost similar kinetics. So it's not actually FIS that drives this cooperative binding, rather it's the deformation of the DNA that drives this cooperative binding. So this bent structure on the DNA, that also is a driving force for protein. But why does that happen? We calculated or estimated the different transport mode of protein, the propensity, and we found that this sliding, hopping, diffusion, all are exactly the same at all the conditions. So how does it actually happen? So what we calculated here is that the net movement, the mean movement of the protein along the DNA, which is average of this dz. So now if there is no target site, then what happens? The mean displacement will be close to 0, right? So that is what we are getting here. But when there is a target site, and if the protein is close to it, so obviously it will sense the target site and move towards that direction, and therefore you have biasness. But when you have bent or deformed structure, or you have FIS which imparts deformation, then you see large biasness towards a specific direction. So it's not really stochastic, it is a biased one-dimensional movement on the DNA that facilitates the search process. We did it for other protein and we got the similar kind of result. So again, we find that this is because of entropy transfer mechanism, where when the protein is not there, there is no net entropy transfer, but when the Phis is bound, you see a sink is formed, and when Gis is bound, then again it neutralizes and then this disappear. And when Phis is not there, there is absolutely no entropy sink that can be observed. I hope this portion is clear. I'll move to another portion, which is DNA supercoiling. This is a critical topology of DNA, which is basically if you consider a rope and you twist the rope too much and then join the two ends and you leave, what will happen? The rope will take a kind of 8-like structure, right? This is called supercoiling, and depending on how it is entangling, that tells you that whether it's a positively supercoiled structure or a negatively supercoiled structure. And this how many times it crosses each other, that will be given by this WR called read. Now this kind of structure is very common in both prokaryotes and eukaryotes, and we wanted to understand how this kind of supercoiling helps protein searching their target sites. So one thing is one should note is that when this this kind of structure happens, two segments of DNA can come close to each other, and then a protein can easily jump from this one segment to another segment. That is intersegmental transfer, we have already mentioned. So we again did this kind of study where we have a open chain, we have a closed chain, and we have chains with different number of juxtaposition sites. So this is again backward master equations we have written for closed loop, and this is for the supercoiled DNA, where with this we have additional term KT, which is basically with the rate that the protein jumps from this portion to that portion, and we calculated the time. So what we found here is that the search time goes down as the juxtaposition sites, number of juxtaposition site increases, which is kind of understandable because juxtaposition site increases, that means intersegmental transfer will be more. The protein will be it's easier to be transported on different part of the DNA, and then it can scan the target site or reach the target site faster. Now if you compare the net increment in the search time with compared to one juxtaposition site and when you have only a closed loop, then and versus you have LD, which tells that you have this juxtaposition site and from this how far the target site is. And this parameter lambda is basically the sliding length, which tells you how long the protein will be on the DNA before it gets dissociated. So if you see, if the juxtaposition site is there and protein jumps, and then it will do one-dimensional diffusion to reach the site. Now if the target site is far, then before it reaching, the protein will again dissociate. And once it dissociates, then basically it loses the advantage of this intersegmental transfer, right? So that means if the target site is close to within this lambda or close to it, then only after intersegmental transfer with one sliding event, it will be the protein will reach the target site. So therefore, as LD is higher compared to lambda, then there is basically no, these two search time is almost similar. There is no advantage. We found the acceleration on it, and we found when the protein does only one-dimensional movement on the DNA, and then there is a huge search advantage. The acceleration is much higher given this plot. So we conclude that this DNA supercoiled topology is a dominant factor, and it does two things mainly. One is basically it increases the intersegmental transfer jumps, which on the other hand opens up another route that can lead to the target site. But when we discuss this, one thing is there is that this juxtaposition site, the movement of that we didn't consider. The second thing is that we didn't consider about positive or negative supercoiling, right? That is also important. So we again did a computational study, and here what we have 336 base pair DNA, and we see that if you impart more twisting into it and then you start the simulation, it takes a nice 8-like structure. So we did it for many structures, and we kind of characterized the propensity that the population of different structures like open circle, open figure 8-like structure, different things, and we found this kind of distribution with different superhelical parameter sigma, and this is more or less matches with the one that people have done, Sarah Harris group in Nature Communication with atomistic simulation. Our structure also matches quite well with the cryo-EM structure. So as we said, we have obtained many structures here. Now, what about the diffusion of protein on this kind of structure? So if you see, we have different architectural protein. So architectural proteins are proteins that is helpful in genome organization. So here we have taken FIS, this protein, and we have find out the diffusion coefficient on the DNA. Now, if you compare on linear DNA, we get the diffusion is much higher on supercoiled DNA. So why that is so? So again, we estimated this probability of different search modes or transport modes of the protein, and we saw that sliding is more or less similar. Hopping is much higher on linear DNA, but if you see the diffusivity of the protein, that is much lower on linear DNA. So why it is so? We found that again the intersegmental transfer plays the major role, right? Because of this kind of juxtaposition site from where protein can jump from one side to another side. And okay, so what we did is now for different this is the negatively supercoiled structure, these are positively supercoiled structure. We found how this D1 varies. We see that it takes a kind of U-like structure. And also we found out that the number of intersegmental transfer nicely matches with that, which says probably this diffusivity is because of this intersegmental jumps. Now, one major observation here you can see. So this is a movie showing diffusion of protein on positively supercoiled DNA, and this is on negatively supercoiled DNA at -1 and +3. Both have one juxtaposition site. Now, if you compare this structure, this plot, you can see -1 you have one juxtaposition site, +3 you have one juxtaposition site, but -3 are higher diffusivity. Similarly, if you see at -2 and -2 and this +4, then also this is higher. -3 is higher than this, right? So all the time we see that in fact for many other architectural proteins we see that the diffusivity of the protein is higher on negatively supercoiled topology compared to a positive supercoiled topology. So why that is so? We found that we measured a parameter called autocorrelation function of this Ci normal vector where at time 0 and at time t, where Ci is basically A cross B and Ai is basically a unit vector connecting the midpoint of the ith and i+1th base, and Bi is the binormal vector. So what does this CTI means? It means that how the twist propagates on the DNA. So it's like you have again a rope, you twisted it quite a lot and then you release one end. What will happen? You immediately see that the torsion that you have imparted into it, it will release from this end, right? So that is something happens here as well. But there is a difference. So if CTI is close to 1, that means that that segment of DNA hasn't experienced any torsion. But if it is less than 1, then obviously there is a twist, and if you see on the positively supercoiled DNA, with this time lag, there is almost no change in twist, right? And if you see the corresponding structure, the juxtaposition site remains almost pinned. In comparison, if you see on the negatively supercoiled structure, you can see there is a huge change, and if you see the corresponding structure within this time period, you can see that the juxtaposition site that is also dynamic and vary quite a lot. And what does that mean? That basically extraposition side move, and therefore the protein also, when it jumps, it will land into different new DNA segment, and therefore it scans the major part of the DNA, and therefore there is a high chance that it will reach the target site faster. So we compared our result with the theory that we have done previously by incorporating the movement of the juxtaposition site, and we found a relatively good match. Another point that we would like to mention here is that the compactness we measured of this structure. What happens is that if you see the negatively supercoiled structure, in presence and absence of protein, the compactness remains the same. Whereas in the positively supercoiled structure, there is a large difference, and that is only for one protein, right? So why this is so? Well, we found again that this is negatively supercoiled structure, and the protein, you can see the red, this is the residence time. So more red means the residence time is quite long, and blue means it doesn't stay there for longer. So on this negatively supercoiled structure, you can see the protein moves very fast, and therefore it doesn't get time really to stick in one place. On the other hand, on positively supercoiled DNA, you can see the protein remains stuck at different positions, and therefore it tries to deform the DNA, because architectural protein, it can impart this kind of deformation, and that is why this kind of structure gets more compact. So we conclude here that DNA supercoiled topology and protein search process for the target site is closely related. The protein that diffuses significantly faster on negatively supercoiled DNA compared to positively supercoiled topology, having identical number of juxtaposition sites, and variation in the protein diffusivity on different supercoiled DNA influences the shape of the latter. Now, why this is significant? Well, this is significant because as I discussed about this topologically associated domain, and experimentally it was found that this topologically associated domain TADs, it can be of positively supercoiled and of negatively supercoiled. Now, when the TADs are positively supercoiled, they are transcriptionally silent. When it is negatively supercoiled, they are transcriptionally active. So we argue that what happens when the negatively supercoiled TAD is there, the protein moves much faster and the communication is much better. So basically that helps expressing the genes, and therefore it is transcriptionally active. On the other hand, when it is supercoiled, positively supercoiled, it is more compact, and the corresponding movement of the protein is not that much, so communication is not that good, and therefore it is transcriptionally inactive. And this work was published in NAR this year. With this, I would like to, so the most of the work is done by Anupam, who is now a postdoc at Rice University, and Sujit and Sangeeta. Sangeeta is here, and I would like to acknowledge the funding agency. Thank you. I'll be happy to answer questions if you have any.
Okay, thanks Arnab. Questions, comments?
No, there is, as I said, it is a stochastic movement. So there should not be any directionality unless there is a compelling reason to move in one direction. For example, the kind of bent structure I have shown, that bent structure can bias the movement of the protein, but that is obviously in a short length scale. For a long length scale, it is almost stochastic. Yeah, so as I said, the major driving force is electrostatics. So basically these DNA binding proteins, they typically have a surface that has more positive charges, so that help it to get associated with the DNA. Not really, because if let's say the positive charge is clustered, then obviously it will get associated, then that binding will be quite strong, and it will not move very efficiently. So the distribution of charge is also important. No, there is no melting. So for example, the pioneer factor that we have shown here, the pioneer factor, their role is typically, as because normal transcription factor cannot bind with the nucleosome, so their role is basically to bind with the nucleosome, open, create a permissive state in the DNA and allows other proteins to bind. Right, there are several opinions. One simple opinion is that they can, kind of compensate the contacts between histone and DNA, and instead of that, they kind of form the contacts with the DNA, and therefore the energetic cost is not that much. But then there is another foreign non-histone protein is already inserted.
Any other comments or questions? I have a question.
Yeah.
That is, see, all through, if I understood correctly, you have discussed search by a protein.
Yes.
There may be some bound ones somewhere in the crowded environment, but there's something that we looked at with your other collaborator, Kolomaisky.
Yeah.
That is, if on the same stretch of DNA, some transcription is taking place. So there will be a RNA polymerase moving.
Yeah.
And then depending on where this protein lands,
Right.
in front or back, the search actually can be hindered,
Right.
so taking much longer, or can be assisted by pushing in that direction.
Exactly.
So that I thought is
So that is important aspect that we didn't consider yet. Yes.
okay. So if no further question, let us thank Arnab and in fact both the speakers of this session.